Assalamu alaikum. My name is Maha Rehman. I'm a PhD scholar in Guwahati University of Science and Technology. Today, I'm going to present one of the objective of my PhD research work, that is on identification, growth kinetics, and evolutionary pattern of spirulina species isolated from freshwater bodies of district Guwahati, Khyber Pakhtunkhwa, Pakistan. These are the outlines of my presentation, which includes introduction, objectives, methodology, results, and conclusion, followed by references. Before getting in detail, I would like to discuss a little about spirulina species. So spirulina is unicellular, alkaliphilic, photoautotropic, and filamentous cyanobacterium, and generally characterized by regularly coiled trichomes. Spirulina have a quite valuable species, and it is known for its high proliferation rate, carbon dioxide sequestration, and high biomass yield. Here are the importance of some of the importance of spirulina species. So spirulina is popular for its antimicrobial activities, including antibacterial and antiviral activities. It is metalloprotective, immune stimulant, have antioxidant properties. It is anti-carcinogenic and boost immune system. Nowadays, spirulina is getting worldwide attention as a dietary supplement for humans as well as for animals. It is also documented as generally recognized safe food by the United States of Food and Drug Administration. The World Health Organization considers spirulina as one of the greatest superfoods on earth. Also, NASA described spirulina as one of the greatest superfood, compact food to travel in space because a small amount contains a wide range of nutrients. So if we talk about the economic importance of spirulina species, so spirulina is the prominent species for dietary supplement worldwide, with worldwide production of 40 million USD annually, and it is estimated that by 2025 the global spirulina market is expected toward 629.6 USD million with a compound annual growth rate of 9.4%. To sort out the taxonomic issues, we find that 16S ribosomal RNA is the best marker because it has a central role in inferring phylogenetic relationships in identification of prokaryotes. This gene possesses most of the characteristics needed for a good phylogenetic marker gene, such as a universal distribution in prokaryotes, functional consistency, relatively large size. Here are the objectives of my presentation. So first one is identification of spirulina strain in freshwater reservoirs. Number 2, evolutionary divergence analysis. Number 3, optimal growth condition and growth kinetics. To fulfill the objective, we followed this methodology. So first of all, we collect the sample from the top and bottom surface of the water reservoirs of Guwahati. So for this, we perform microscopy and then enrich the sample. After that, we transfer that sample to the lab aseptically. For isolation, we followed through two different methods. One is the pour plate method containing agar media with BG11, and the other one is that we only use BG11 media without agar. Then after each 24 hours, microscopy were performed to check the culture. For further confirmation of our species, we perform molecular identification using 16S ribosomal RNA marker and then move towards the growth kinetic experiment. So for this, we optimize the condition for comparative analysis at different pH, temperature, and light intensity. Here are the results. You can see in the map that we select the city of Kohat because Kohat is the main district of KPK Pakistan, and the presence of limestone and heavy metal in the mountains of Kohat make the water more alkaline in nature, which is suitable for the growth of spirulina species. So we chalk out some of the water reservoirs of Kohat. Some of the names are mentioned here. That is Tana Dam, Bona Sharif, Jungle Khel Chashma, Khandar Dam, Jarma, Ghandiali Dam, Dhoda Sharif, and Chili Bagh. So here we mark the area of Chili Bagh, Kuhar, red in color because the first report we get were from Chili Bagh, Kuhar, which is the area in Ustarzai. Here is a microscopic picture of spirulina species. Here are the results of DNA isolation and PCR product amplification. This is the phylogenetic tree for spirulina species. Here are the growth optimization results. So growth of spirulina in response to varying temperature condition. So the best results shown were at 20 degrees centigrade. Here is the result of light intensity, growth of spirulina in response to varying light conditions. So the best results were shown at 2400 lux. And growth of spirulina in response to varying pH conditions. So the best results shown were at pH 10. Here is the result of growth rate calculated from measuring optical density and weight of spirulina species per ml for DG11 media for a period of 12 days. So the maximum growth we get were at day 10. Here are the conclusion of my presentation. So first one is that spirulina is a non-toxic balanced diet with high concentration of protein, vitamins, and essential fatty acids. Number two, the development of spirulina in the lab has been investigated under various settings. Number three, the best spirulina growth yield was achieved at pH 10, temperature regimes of 20 degrees centigrade, and light intensities of 2400 lux. The experimental results allow for accurate anticipation of biomass production for large-scale spirulina cultivation with higher photosynthetic content. Here are the references of my presentation. I cordially acknowledge and gratitude to Professor Dr. Rehman, my supervisor, Professor Dr. Nur Muhammad, Chairman of Department of Biotechnology and Genetic Engineering of Bharat University of Science and Technology, and special thanks to the Higher Education Commission of Pakistan for funding this project. I really thankful for the IBBC 2023 for giving me the opportunity to present my research work here. Here I mention my email address. If somebody have any question, please inbox me. Thank you for listening. In that.